So like I said, next up, I'd like to introduce Wenbo Ma, professor of plant pathology here at UC Riverside and co-inventor of a patent that can have a direct impact close to home. So Wenbo and Georgios have developed a technology that can help detect a particular citrus disease, HLB, that has decimated the Florida citrus industry and is threatening California's. So I'll let Wenbo fill in the rest of the details. Wenbo, thank you very much.
I would like to really thank Brian and Larry and Mike and everybody in the Office of Research and APAC for giving me this great opportunity to talk about our patent. And it's really, I'm not used to this kind of audience, so my presentation will be quite research-oriented, but hopefully this is big enough for you to see. What our labs are, me and my research collaborator, Georgios Vidalakis, who is also a professor in the Department of Microbiology and Plant Pathology, what we have been working on is this very severe citrus disease called Huanglongbing. It's a Chinese word. It translates into English as yellow shoot disease, and that's one of the symptoms of this disease. But first of all, I would like to say that citrus production is very important to the U.S., and this is citrus producing map, and you will see the darker color are the main citrus producing areas, and the U.S. is the third, just after Brazil and China, and a lot of it is produced in California. But this industry is severely threatened by this one single disease called HLB or Huanglongbing, and here you can see some of the disease symptoms, and obviously the fruits that are produced from infected trees is not marketable at all, and that's very important for California because we are the main producers of table fruits. During the past 10 years, this disease has caused a really enormous damage to the Florida industry. And this is the same orchard that was pictured just 1 year and 1.5 years apart. And you can see from a healthy looking orchard and turn into an orchard filled with dead trees. And there's nothing we could do about this disease. So I hope you can see, and this is very dramatic figure, and this is a citrus production in the last 10 years in Florida, and you can see the reduction. This is 70% reduction. This is really huge damage. And this is another way of looking at it. That's every year in Florida only, the citrus industry lose almost $1 billion cash. So this is really, you may not be aware, but this is a huge damage. And the problem of all that is that there's no cure. So once the trees are infected, there's nothing anybody can do except for watching them to die. So eventually, you will see orchards like these. The farmers can only chop them down, and then when you regrow new seedlings, they will be infected again. So the cure is one of the problems, and the other problem is diagnosis, obviously. So I'll talk about our patent later on. And I found this just came out recently. I found this very interesting. This year is the first time in history that the California citrus production surpasses Florida citrus production. And that's only because Florida citrus is damaged so badly by HLB. But we shouldn't be happy because HLB is here too. So California, let me show you this. This is the title of a news article in the PE, and that was published in July 26th. So that's about a month and a half ago. That was a tree was identified to be HLB infected in Riverside, close to campus. So actually, UCR now is a quarantined area by USDA. And is it really important? Of course, because Riverside, we have the first citrus trees of all the United States. And if you can see that historical picture there, that's actually a Mission Inn, and the gentleman in the middle who was replanting one of the two parent citrus trees is President Roosevelt. So this Riverside really has a very significant long history of citrus production, and one of those two trees is still alive in our citrus historical park. But we may lose all that if we don't cure this disease. As I said, there's no cure. So very simple introduction of the culprit is caused by a bacterial pathogen called Liberobacter candidatus liberobacter. In the US is a species called Liberobacter asiaticus, so we call it Clas. And this bacteria has an insect vector, so it's either in the insects or in the citrus trees. And the insect, when they feed on a infected tree, they'll pick up the bacteria, and then when they feed on a healthy tree, they'll pass that bacteria to the healthy tree. So if we have infected trees in the field and we don't know, and this insect will spread the disease out. So, and without any curative methods. So all we can do is to remove the infected trees so that they won't serve as source for further infection. And that really makes the diagnosis very, very important, because these diseases are really this particular disease is very hard to be diagnosed because these bacteria are hiding in the phloem. So this is the vascular system of trees. And they have very active material transportation in there. But this also gives us opportunity because maybe we can detect some things that are circulating and then spread out in the system. So that's the basis of our patent. So, oh, I forgot I have this slide, and this is just to show you the one on the right with these plates with holes, and these are the phloem system. And you can see that because it's not really a tube, it has this septa, and the bacteria cannot move very fast in there, or many times they don't even move. So if you imagine this big tree, and this infection may only happen in a couple of branches, and the bacteria will stay in that branch. But if you take a sample from another branch that without the bacteria, then you wouldn't think this tree is infected, whereas it's already infected. So what our approach is to identify a protein biomarker, and these proteins are small enough so they can actually go through those holes that in the phloem system, and then you would be able to detect it from in a branch that doesn't have the pathogen. Okay, so that's really the whole basis of our patent, and we found a very good biomarker, and it's small enough, and it's secreted by the bacteria which are in the phloem, and once the proteins are secreted out, they are actually travel and distribute in the other part of the tree, and that really enhances our chance of getting a sample of this tree and then get a positive reaction by using an antibody-based detection. So this an antibody we can generate from animal models, and then these kind of high affinity binding to the biomarker, finding them, and we can see that reaction if there's binding going on. And that would give us a positive diagnosis. And there's a diagram showing there, and what I really want to emphasize, the advantage of this patent is that these proteins can move beyond where, and be present beyond where the pathogen cells are, so that the PCR or DNA-based detection would come up negative if you didn't really take that branch containing the bacteria, but our method will become positive because the protein can move faster and spread wider compared to the pathogen. So, so we do have some very nice results, and this is just one of the evaluation assays that we did. We took 108 trees from Texas. Texas has about 30% infection for this disease, and then we can compare that to the only standard diagnosis, which is PCR-based, DNA-based detection. And what you can see there is that for the PCR positives, we also have a very high positive rate. It's not 100%, so we're still improving, but the vast majority is that we also got positive. But from the negative trees that PCR came out negative, we can diagnose more potential positives from these would be false negative trees. So I'm not going through the details, but we do have evidence showing that our method can behave better in certain cases than the nucleic acid-based detection. And then eventually what we want is to have a kit, and it's called ELISA kit, and using antibodies, and we can provide this to the customers, some reagents and some antibodies, and then these reactions can occur on these 96 well plates. So these are very small plastic plates with 96 wells, and then we can, in one run, we can test 96 different samples. So this is rather fast and cheap and high throughput. And so this is the first step, and now we already have one in place. And the second step is that we can improve and having more sensitive ELISA assay by introducing more antibodies in there. And the third step is that we could further modify this test by producing like pregnancy paper type test that people can bring to the field and directly test their tree. So, my collaborator, Georgios Vidalakis, he is an expert in citrus diagnosis, and he developed assays for other diseases too. And so I put the title is, there are many other diseases that damage or threaten the citrus industry in addition to HLB, and many of them are viral diseases, and Georgios is an expert in that. And all these diseases also cause potential huge damage to the citrus industry. So I'm not going through the list. I'm just saying that Georgios has developed this multiplex test. So using one single test, he can detect 6 different diseases, and another single test, he can detect 14 different diseases. So from 2 tests, he is able to call positive or negative for 20 different diseases. And this is really very powerful and also very high throughput. So with that, I wanted to conclude and thank all the people in different departments at UCR, and especially Brian and Joyce for the IP, and James for industry relationship, and lastly but not least, Larry and Ken for really helping me to really grow into a potential candidate entrepreneur, and I'm learning a lot. It's a pretty steep learning curve, and of course my collaborator Georgios, and also my postdoc Agustina Di Francisco, and Deborah Pagliaccia there, and also PhD student Kelly Clark, and this research has been funded by USDA and the Citrus Research Board, which is the California Citrus Commodity, and also by UCR Proof of Concept grants. Thank you very much.